Hello, my name is Nain de la Cruz, and together with William Albornoz and Jesus Rodriguez Gonzalez from Simba staff at Monterrey, Mexico, I'll be presenting our work on H infinity and structure uncertainty analysis of p53 signaling. And specifically, this work shows how robust the cell's decision to undergo apoptosis really is. The outline of our presentation is as follows. First, I'll start with the biological motivation. After that, I'll walk you through the mathematical model we built and linearized. Then I'll cover the robustness methodology employed for the research. And finally, we will show the results, their biological interpretation, and the conclusions. I would like to start saying that cancer remains one of the biggest public health challenges we face. For example, projections put annual cases at 35 million by 2050. A central player here is p53, also known the guardian of the genome. When a cell is stressed, p53 activates cell cycle arrest, DNA repair, or apoptosis, depending on the DNA level damage. It's so important that p53 is the single most frequently mutated gene in human cancer, with functional loss in over half of all tumors. p53's activity is controlled by its antagonist, MDM2. MDM2 is an enzyme that binds p53, blocks it, and tags it for degradation. However, on the other hand, p53 also activates MDM2 transcription, so together they form a negative feedback loop. This loop keeps p53 low under normal conditions, but lets it respond quickly to stress. When damage is severe and irreparable, this same loop can sustain p53 at a high stable level, committing the cell to apoptosis. The problem is that this loop can be disrupted. MDM2 gene amplification shows up in roughly 3 to 4% of all human cancers, and it suppresses p53 activity, letting cells evade apoptosis and resist treatment. So the question we wanted to answer here is under what parameter variations, from mutations or just normal biological variability, does this apoptotic commitment actually break down? To answer that question, we borrow tools from robust control theory, more specifically H infinity and structure singular value or mu analysis, which are tools that have already been used successfully in biology. For example, they have been employed to study the robustness in gene regulatory networks against molecular noise, also to design robust closed-loop insulin delivery for type 1 diabetes, and more broadly in biochemical pathway models. These studies show that the robustness margins from this framework are biologically meaningful, and they connect directly to how well a system keeps functioning under real variability. Although these tools have been applied in biological contexts, they haven't been used to analyze the robustness of the apoptotic decision itself at the intracellular level. And that's the gap we want to address here, characterizing how robust the apoptotic state is to parametric uncertainty. From this work, we expect two main contributions. First, a structured singular value analysis that identifies the specific combination of parameters that pushes the cell out of apoptosis. Second, we quantify the gap between what the linear robustness margin predicts and where the true nonlinear system actually loses stability. In this slide, we show the mathematical model, a graphical representation of the mathematical model we employ for the research. It shows four different variables: p53, MDM2 mRNA, MDM2 protein, signal damage D, and an external input U. The block shows the interconnection between them, and arrows in the diagram mean production or activation, while T-bars mean repression. The graphical model was translated into a set of four differential equations, ordinary differential equations. And in the first equation, we see p53 production, which is repressed by MDM2 through a Hill function, and the degradation of p53 follows Michaelis-Menten kinetics, with the damage signal directly scaling the degradation rate. And the second equation shows MDM2 mRNA interaction. In here, the mRNA is activated by p53 and degraded by a saturable enzyme. In the third equation, we see the MDM2 behavior, the protein behavior, and we see that it's translated and degrades linearly. And finally, the fourth equation shows the damage signal, which responds to a step input and decays on its own. In the following slide, I showed the parameters we employed for the equations shown in the previous slide. And these parameters were fed to obtain qualitative behaviors of the interaction between p53 and MDM2. This behavior is shown in the following slide. With a moderate damage input, the model produces sustained p53 oscillations, which is shown on the left of the screen, with a period of about 6 hours. That's the repair regimen. If we increase the damage, then we instead see damped oscillations settling into a stable high p53 level. This is the apoptosis regimen. Both results match experimentally observed p53 dynamics. Our robustness analysis is centered on the apoptotic fixed point, at which the value of u equals 2. So when we find the fixed point, then we obtain the following results shown on the screen for p53, MDM2 mRNA, and MDM2 protein, and the damage signal. With this fixed point, then we linearize the system around it and obtain the Jacobian matrix A. We also find the eigenvalues, which all happen to have negative real parts, so we see that the fixed point is locally stable. This confirms that apoptosis is a genuine stable state. But we also have to notice that the complex pair has imaginary part corresponding to a period of about 4.3 hours, which is close to the natural repair oscillation period. That's a resonance, and it turns out to matter a lot for robustness. Now, the robustness question. We treat 6 kinetic parameters as uncertain, the ones most consistent with observed cell-to-cell variability, K sub 1, B sub M, D sub 3, delta, K sub D, and delta sub D. Each of these parameters is allowed to vary up to 30% of its nominal value in either direction, and together they form a 6x6 structure uncertainty block called delta. To analyze this formally, we write the perturbed Jacobian as a linear fractional transformation separating the nominal system from the uncertainty block delta. The interconnection matrix M sub 11 captures how the uncertainty fits back into the system. The input matrix brings in the damage signal, and the output matrix selects p53. This structure is what lets us tie the analysis directly to specific biological meaningful sources of variability. The first tool we use is infinity norm, which is the worst case amplification of a disturbance across all frequencies. In our case, it tells us the worst case amplification from a damage signal disturbance to p53 output is a standard, well-established robustness measure in control theory. Although H infinity treats uncertainty as arbitrary, any perturbation of the right size can be found by H infinity. However, the structure singular value, mu, is more precise. It restricts the uncertainty to our actual 6 parameters structure. That makes it less conservative, and critically, it tells us not just how much uncertainty destabilizes the system, but which combination of parameters does it. If we take the inverse of the maximum value of mu, then we find a single number called rho, which is the robustness margin. If rho is greater than 1, then the system is robustly stable within the predefined 30% bound. If it's less than 1, then some combination of parameter variations within these bounds is enough to destabilize the system. Although all of the analysis was implemented in MATLAB 2024B using the Robust Control toolbox for H infinity and mu analysis, and the nonlinear validation was carried out using the ODE 45 function of MATLAB with tight tolerances. The first result is the H infinity norm. which has a value of 2.383, peaking at a frequency of 1.47 radians per hour. We have to remember that this is close to the resonant frequency. Once we introduce the 30% bound, the uncertainty bound, then the worst case gain jumps to 5.61, which is more than double. The peak frequency barely shifts, which tells us uncertainty is amplifying sensitivity and that the same resonant frequency is kept and not moving elsewhere. On the other hand, the mu analysis tells us a more precise story. For example, the upper bound peaks at about 0.49, again near 1.47 radians per hour, and stays below the critical value of 1 across all frequencies. That gives us a robustness margin of 1.757, consistent with what we will expect from the mu analysis. But what does the raw value mean? It says the linearized fixed point is robustly stable within our 30% bounds. And in fact, we will need to scale those bounds out to about 53% before the linearized system becomes unstable. We also identified the specific worst case parameter combination at a destabilizing gain corresponding to 72.3%. So the true linear stability boundary sits somewhere between those two numbers. This table shows that a specific worst case combination, all six parameters move together. For example, k sub 1 and delta sub d increase, while b sub m, d sub 3, delta, and k sub d decrease, each by the same 72.3%. This combination isn't an arbitrary set of deviation. It's the exact direction the mu analysis identified as most dangerous. The results obtained before with H infinity and mu analysis is confirmed by a nonlinear simulation. In this case, we have the question, does this worst case direction actually matter for the real nonlinear system? And so we took all the 6 parameters and made the variation within the bound of 30%, and we find that if we keep the system or if we keep the parameters within the 30%, then the apoptosis is kept for the system. However, if we pass the 30%, then the system goes into the oscillation state, indicating that the cell cannot anymore reach the apoptosis state. So, from these results shown previously, the central findings are the follows. The linear analysis said we need up to 53% of deviation before losing stability, but the full nonlinear system already loses stability at 30%. But we will ask why there is a gap between the linear result and the nonlinear result. Well, since we are doing linearization around the fixed point, then the operating point has been fixed already. However, in the nonlinear system, the operating point itself shifts as parameters change. And this is an observation that has been made already in classical results from Khalil, for example, or from Gockenheim-Meyer and Holmes. Local linearization is only valid near equilibrium, and even worse, that neighborhood shrinks as you approach a bifurcation. So the takeaway here is that the raw value we found should be read as an upper bound on the true robustness, not a direct threshold for apoptotic evasion. So to summarize, the mu analysis correctly identifies the parameter direction that drive apoptosis evasion, and also we quantified the gap between the linear model and the nonlinear model. It is important to keep in mind that the variation of the parameters must be in the direction and at the same time of the mu analysis gave. So it is important also to highlight some caveats. In this case, the robustness margin is strictly a property of the linearized system around the fixed point, and mapping this parameter direction onto specific oncogenic mutations will need experimental validation. Also, from this observation, it is important to note that It is necessary to conduct a nonlinear robust analysis using nonlinear robust frameworks in order to have a more real and more meaningful result for the p53 MDM2 interaction. And with that, I would like to thank you for your patience and for seeing this presentation. And if you have uh, any question, or you would like to make any comment about the presentation, all comments and questions are welcome to the following address shown to you in the presentation. Again, thanks a lot and have a good day.